Can you see my screen? So I imagine you can. So good, excellent. Then let's move on. So my name is Silvio Rizzoli. I'm a professor at the University Medical Center Göttingen in Germany, and we do a lot of imaging. You know, I graduated using electron microscopy. I moved into biochemistry and then into super resolution fluorescence and did a lot of imaging. And there are lots and lots and lots of imaging techniques one can use. I mean, on the right-hand side, you see some beautiful results from my lab that reflect active motion of molecules in a small volume, you know, in a volume of something like about 2-3 micrometers in width. Here you see a 3D reconstruction on the left-hand side made. This is a static picture, as you of course can see, showing you how organelles and proteins are organized in synapse. Again, this is on the right-hand side, you have a presynapse. On the left-hand side, you have a postsynapse. These are the elements that talk in the brain. You know, you can do a lot with fluorescence imaging. You can do a lot with electron microscopy. You can do a lot of biochemistry. You know, there's plenty of beautiful techniques out there. You can get really, really nice pictures of what life looks like. But there's one problem you can't really solve with all of these beautiful light-using technologies, and that is things going on over longer time periods. So what's going on, let's say, over weeks? Let's start with the following problem. You know, I want to give you an example. Let's start with that. So you may have heard of erythropoietin, because, for example, in the Tour de France, often the cyclists, at least in the late 90s and 2000 years, the cyclists were often taking erythropoietin as a doping drug. You know, long distance runners do it as well, or at least did it until about a few years ago. Now it's very much tested, you know, to see if people are actually taking this, but it is still quite used. It's also used in medical practice because it improves your blood flow and heart performance. And people have discovered a few years ago, about a little more than a decade ago, have discovered that it also improves mental performance. So what you can do is you can take mice, you can inject them with erythropoietin at different times, and remarkably, they get smarter. Here you see on the right-hand side, you see a graphic that shows you how fast mice can actually do a task. And the erythropoietin injected ones do it faster. You know, they learn faster and they do this task faster. The normal mice take a lot longer. So what does that mean? You know, you inject erythropoietin in a human or a mouse and they get smarter. How? How is that? What does this do to the brain? Do the humans and mice get new neurons? I mean, you know, that's the easiest way to get smarter. You make new neurons. You know that neurons are born very infrequently, and it's not even clear if in humans they get born at all. In mice, it's also quite difficult to know what exactly is going on. So how do they get neurons? Do they get them? You know, what is going on when you inject erythropoietin in the brain of a mouse? You would need to analyze changes over weeks and months. And this you simply cannot do with fluorescence microscopy. You know, I can try certain things, but it really is difficult and it doesn't really work. What you would like to do is to do what is called isotopic labeling. For isotopic labeling, this is called the SILAC approach. You take your mice and you let them eat food that contains heavy isotopes. So something like carbon-13, nitrogen-15, and so on. They eat the heavy isotopes, and new cells, like new neurons, would incorporate these heavy isotopes. Normally, then you take the brain, you homogenize it, you make all sorts of biochemical fractions out of it, and you use mass spectrometry to analyze it. You know, and this is then what you get. You know, you get the turnover, you get the long-term changes in these animals, and we know about this. You know, this is from one of our publications. We know all about this, but it's in solution. You know, this is a mixture of proteins. You cannot tell where those molecules are coming from, which neurons are behaving how, and so on and so forth. I mean, you know, this is a very difficult thing to do in terms of imaging. But what if there was a machine that actually allows you to image the isotopes? So you have your mice eating this isotopic food, it gets incorporated in specific cells, and then you could actually see them. And as you, of course, most likely know, there is such a machine, and that is a NanoSIMS. And the way it works is you have a primary ion beam that is going to encounter the surface of a specimen and is going to explode, so to say, particles out of it. The charged particles, like ions, they are going to be then measured by detectors, and you have this beautiful picture here that shows you the ion source is sent onto your specimen, which could be a neuron, for example, and then your secondary ions are coming out and are being collected by ion detectors. And you can do 7 different ions at the same time, I mean, which is a very, very important way of measuring. And then you could actually look at what you fed the mouse with. How good does it work? You know, for a biologist, the first question when I look into such a machine is, well, how good is the resolution? What can you see? And in this example, this is from our first publication with NanoSIMS. You see here little balls of gold. They are little gold particles, and we are simply measuring them. And then we are doing a line scan across. And you see the full width of maximum of this line scan, which is a measure, which is mostly like a fluorescence-based measure, to be frank with you, that tells you the size of the objects. This is about 50 nanometers. If you use a measure which is more adapted to the fact that this is not fluorescence, this is a mass measurement, then it goes below 50. It goes to something like about 30 to 40 nanometers. But we take a very conservative approach here, and I would say on a good sample, you are somewhere around 50 nanometers, which is quite good, to be honest. I mean, you have to think the smallest organelles of the cells are also around the 40, 50 nanometers. So this is pretty good. Depending on what samples you look at, you know, here we are looking at 14N, here looking at 15N, both in the form of Cn minus ions, depicted here as 14 and 15N. Here you get an apparent resolution which is a bit higher, basically because these are cells, these are real things, and they don't have sharp edges. A gold particle has a very sharp edge, so you get a very nice resolution measurement. Biology doesn't really have sharp edges, or very rarely, and then your apparent resolution is a bit lower, but probably your real resolution is probably quite okay, and is probably quite in the direction of 50 nanometers. If you go in the z-axis, it gets a lot better. So in the z-axis here, we take many pictures of the same sample, and in the z-axis, you basically measure how is my sample changing, you know, along the z. And from that, you can determine resolution. And that's somewhere between about 5 and 15 nanometers in a biological sample, which is, I would say, extremely good. I mean, that is just exquisite. You know, this is, it's hard to get it better. You know, in biological imaging with fluorescence, for example, you rarely approach this. I mean, this is really, really good. So, what else? Well, you have good resolution. You have a very nice resolution in the X, Y, and in the Z. But how do you know what is what here? You're looking here inside a cell, but what part is what? That's a bit of a problem. How do we know what each structure is? This is probably one of the most difficult things of the secondary ion mass spectrometry technologies. You would like to also know the identity of the things you're looking at. In fluorescence, what you label is what you see. So you already know your identity because you just label the things. In electron microscopy, you see the shapes very well, so you kind of interpret from those. In SIMS, this is a little more complicated. So sometimes you have an obvious, you have an obvious sort of identity. So here we are looking, in this particular example, we are looking at heart muscle, and you're looking at 1, 2, 3 microcapillaries in the heart itself. This is heart muscle, these are the capillaries. And if you look at 14N, you see 1, 2, 3, 4, 5 little cells in here. If you look at 13C, and these were mice fed with 13C for about 2 weeks, what you see is 3 cells very prominently, and 2 of them, this one and this one, you don't see them at all. What does that mean? These are all erythrocytes. These are red blood cells. The ones marked in blue are old. They did not get any C13, so they are old ones. The red ones are young erythrocytes that were made with a lot of C13. And then you see them, if you do a ratio of these ions, you see them very prominent, the young ones, and the old ones, not much at all. So, you know, this happens, and sometimes you have very obvious things that you immediately can tell what you're looking at, but it's not so common. You know, so ideally you would like to label somehow to know where your samples are. How can you do that? Well, there is one possibility that is actually quite simple. Do whatever treatment you would like to do to your mice, then dissect the elements of interest, put them in a plastic material, cut them, and then look at them in NanoSIMS, and look at similar samples, maybe not exactly the same sample, but look at the next cut. So if you cut a thin specimen, you embed your mouse brain, let's say. In this case, you're looking at retinas. You embed the mouse retinas in plastic. You cut them in a thin section. Then basically you are going to use this plastic section for imaging in SIMS. The next cut, the next section of the plastic section, you can put on another carrier, like a glass slide, and then you can stain it in histology. So you can get sort of like a toluidine blue or some other histological stain, and then you can tell about the morphology of the different cells. And you can say, well, this is what it looks like, so I know this is what it is, so on and so forth. You can kind of tell the identities of your cells. Works very nicely for cells. It doesn't work for the, let's say, small organelles inside the cells. You know, it's hard to correlate. So what else could you do? But, you know, a take-home message, you can use histology quite nicely to correlate with SIMS, and then you get the identities of the cells. And often this is all you need. But if you want to go deeper, what you want is correlation with microscopy. And what you see here is the first example that we produced of what is called, what we called COIN, correlated optical and isotopic nanoscopy. So essentially, you have your sample, which is fluorescently labeled. In multi-channels, you can get also super resolution to work here. And then you take the same exact sample and you image it in the NanoSIMS. And then you see, in this particular case, we could actually look at different organelles. These different colors are marking different organelles. And then the neurons have been fed with N15 isotopes. And then we can look at where these N15 isotopes are localized in the neurons. And as you can see, for example, this green label sort of has a certain shape. And you see here, this is a ratio between N15 and N14, telling you where the newly made proteins are. And you see it kind of follows this green label. You know, it's somewhat similar. And this you can get in a measurement and blah, blah, blah. You know, I mean, I don't want to bother you with statistics and whatnot, but it gives us an idea about how the cells are able to process their material over long time periods. What else do you need to do for this? Well, it's not such a difficult procedure. So you just have to grow your cells for your animals in presence of whatever isotopic metabolite you want, like N15 leucine. In our case, leucine is a very important amino acid. With N15, then it gets incorporated in all newly made proteins. It can show you where the newly made stuff is. Then you do your fluorescence imaging, labeling however you want, immunostainings, affinity labelings, whatever you may like. You embed the samples in a plastic resin. You cut them with an ultramicrotome in something like typically about 200 nanometers. Then you image them in fluorescence. Here are your samples. They are placed on a silicon wafer. You know, this is like a non-transparent, hard cover slip. You image these samples on a microscope, and then you may burn sort of markers with a laser around the sample to know exactly where you are. We realized over the years this is completely unnecessary, so it's kind of like optional. You don't need it. You can identify the sample very nicely in the NanoSIMS again, and then you identify the sample, as I said, in the NanoSIMS and image it in the NanoSIMS. You know, it's really what we didn't know when we started this, 8 years ago. We thought you really need a marker to tell you exactly where you are. We learn over the years, it's very easy to find the same cells. Well, it's not very easy. It is, though, quite feasible to find the same cells in NanoSIMS. looking just at the shape of the cells. And once you found them, the general outline of the cell and the autofluorescence of the cell in fluorescence imaging can be overlaid beautifully with the carbon or nitrogen signals in SIMS. So you really don't need any other type of markers. You can do them, but they are not necessary. You can very, very nicely overlay your cells. What can this be used for? Well, you can use these neurons that you feed with leucine, and you can look at, for example, synaptic vesicles. Synaptic vesicles are things that release neurotransmitter and communicate stuff in the brain. And there are some ones that are active and always communicate, and there are some that are called the reserve, and they are never communicating. And then what we did a few years ago, we basically labeled the neurons with leucine. We identified in fluorescence the different types of vesicles, and then we just overlaid. We overlaid these type of pictures in the NanoSIMS, the ratio of leucine of old and new isotopes. We overlaid this with the fluorescence images, and then you can start to see the reserve vesicles typically overlay with bluish sort of spots, which tell you they don't have a lot of young proteins. The recycling vesicles were overlaying with plenty of spots where there was young protein, telling you our synapses in our brain use young vesicles. It kind of makes sense. If you want to have very good synapses, you want them to use young proteins and to be very sort of clean and fresh. And the older vesicles you kind of push into retirement. So it was not known at the time, and honestly, there is no other technology that you could use to actually tell you this. And it's a very interesting finding. You can do more. You can look at synapses in different physiological conditions. You can modulate synapses. You can do multicolor here imaging of all sorts of things in synapses. I'm not going to bother you with the details because that's what they are, just details. You can do a lot more and then correlate with, in this case, was again N15 labeling, and you correlate with this, tell about how specific synapses work. For example, here we published last year the conclusion that synapses that work very hard, they get more proteins. It's synapses that work hard, they get supplied with new proteins so that they can stay nice and working well. And synapses that do not work hard don't get proteins, which is kind of a crazy conclusion. Nobody knows how this happens, but it does happen. It's again a very interesting new finding. So what else can you do? You can go inside animals. You can go here. We are looking at the inside of a brain of a mouse that was fed this time with C13. You can get your fluorescence labeling. You see here different types of fluorescent markers, and then we can correlate this with the ratio of C13 to C12. So wherever we see a high ratio, that means there is a lot of new protein that has been incorporated. Where we don't see high ratio, well, there's not much new protein. And this, we can do statistics and analyze and everything. And this taught us that there is a new type of brain plasticity going on that people didn't know about before. That there is that we call it the recycling of the extracellular matrix. Again, the extracellular matrix is sort of like as you hear it, a matrix is sort of like a meshwork that keeps our brain cells together, and its dynamics are very hard to understand. And this gives us a new clue on how these dynamics work. And again, you couldn't do this with fluorescence. There is no way. You had to feed the animals with something for weeks and then see where it got incorporated. Good luck doing this with fluorescence. There is really no way. So now perhaps you still remember the problem that we started with. We wanted to understand this erythropoietin doping. So this we doped the mice with erythropoietin. It improved mental performance. So did they get new neurons? What happened in the mice? So now finally with this correlation with fluorescence, we can finally ask this question, and let's see what happened. What we have here is the hippocampus of the mice. This is a region which is very important in the memory of the animals. So memory and learning and understanding and remembering new things. And we look at it in green, we see the cell bodies of neurons. And in orange, this is, you see here the letters EDU. in orange, this is a labeling that shows us newly made cells. So you look at the main part of the hippocampus, and there are very, very, very few newly made cells, almost none. And if they are most likely not neurons. So did these animals get new neurons? This is all fluorescence labeling. And if you just look at the fluorescence labeling, you would say, not really, because, well, we don't see new neurons getting born. Now let's do the experiment with the NanoSIMS. We take these same animals and we feed them with an N15 diet. So wherever we see more N15, we would say something happened. If we do a placebo-injected mouse, so one that was injected with some sugar, it's a fairly homogeneous picture. You know, the ratio of N15 to N14 is fairly homogeneous. You see here, these are the nuclei of the different neurons. These are the nuclei. They all look kind of the same. If you now look at the erythropoietin injected mouse, what you see is a very bright one cell. You know, we zoom in here and you see this cell has a somewhat brighter nucleus, but also very bright spots inside the cytosol and inside the neurites that are just moving left and right. So what happened to this cell? It just got a tremendous amount compared to the other cells of N15 all of a sudden in it. Well, what that means is that this was a cell that was probably dormant. You know, it wasn't doing much before we did the erythropoietin. And then once we injected it, it just made a lot of new protein and just put this protein in itself and grew and got to be a large neuron. This was most likely a small precursor cell, and through the injection of erythropoietin, it got to be a big neuron, and it has now its nucleus colocalizes with neuronal labeling, and as you see, there is no orange labeling. So it definitely, this was not a cell that divided to make itself into a neuron. It just grew. It just grew inside the brain of that mouse to become a neuron. Now, what technique can you use to show you that a cell grew inside the brain of an animal? I mean, honestly, other than NanoSIMS, I know nothing else that would actually do this for you. So, of course, you can analyze this, and there is a population of neurons that did this and blah, blah, blah. And essentially, what we concluded was that erythropoietin treatments induce the formation of new neurons in the brain from old precursors, from old dormant small cells that just grow into these nice big neurons with little or no cell division happening. So it tells you it's a very, very intricate and very interesting phenomenon, very interesting mechanism, that you have some little cells in your brain that are just waiting to be activated, and the erythropoietin activates them. Now, should we all start to inject erythropoietin into ourselves to become smarter? I certainly wouldn't mind being smarter than I am, but it's dangerous. I mean, I wouldn't do that because this type of mice also get at some point too many neurons and end up with epileptic fits, because at some point there is too much excitation in their brains and they get very sick. So, of course, this is not something that humans would want to do, but nonetheless, it's very interesting that an element that normally modulates the blood vessels and the heart has such a profound effect on the brain. What else does it do? You know, over the years, we looked with our collaborators from the group of Hannelore Ehrenreich, also here in Göttingen. We looked more into this, or rather, they looked and we helped them. And I won't go into details, but another conclusion is that erythropoietin changes the balance between neurons and microglia. So essentially, microglia, which are the immune cells of the brain, they become differently modulated and they behave differently. And this is something, again, quite intricate. Let's not go to every little detail, you know, because I don't think you want all such details. But long story short, inject the erythropoietin into an animal or a human, and probably very strange things will happen to their brain, not just to their cardiovascular behavior. So, what else can you do? You can correlate now, instead of fluorescence, you can correlate with electron microscopy. That's just beautiful. I mean, these are the most beautiful correlations, because you can cut your specimen, have it labeled for electron microscopy, image it, and then look at it It's the same exact specimen in NanoSIMS. And you see here some examples. And of course, you can analyze this in many ways. In this case, what you're looking is at myelin. This is like a protective coat that we have on the axons. And in this particular paper, our conclusion was that different regions in myelin have different characteristics, and this is a very, very interesting conclusion. And some regions make a quick sort of change, they grow quickly, and some regions are a lot slower in growing. So that's again, let's not go to too many details, but it's possible to do this correlation. Then another type of correlation that is quite nice is triple. So let's do all three. So let's do fluorescence, electron microscopy, and then let us do the NanoSIMS. So you do fluorescence first, electron microscopy second, and finally the NanoSIMS. And you can do this. You can embed the specimen in a plastic, and then you can process it in thin sections, then image them on an electron microscopy grid, imaging fluorescence, then takes them to the EM, then takes them to the NanoSIMS, and then you can correlate all of these types of information. What can you do with this? Well, you can use fluorescence for a functional analysis, electron microscopy for a structural analysis, and NanoSIMS for example, for looking at the isotopic composition. And this is an example, you see, in this case, mitochondria are labeled here in light microscopy. You see the SIMS picture, you see the electron microscopy, and then you see the overlay. And you can learn about mitochondria that are not labeled in fluorescence, meaning they are inactive. So you see an inactive one and you see some active ones, and they have a different isotopic composition, a different basal fundamental isotopic composition. It's completely different. And this is something that nobody would have predicted before, but it's actually easy to see once you correlate. So these are very simple fibroblast cells. And just looking at the very fundamental basic composition, it's amazing. It's actually different from an organelle to another, just because this one up here is not active and this one down here, it looks exactly the same in electron microscopy. This one is active, and then it has a different atomic composition. Fantastic. Now, so far so good. You have all of these nice correlative methods. The question is, can we get better? Correlation is nice, but it does require a lot of work. So maybe we could label directly into the SIMS pictures. You can do that, and our group has over the years made a few probes, for example, containing fluorine. So here we have a fluorine probe that labels the endoplasmic reticulum of this cell, and you can actually see it very nicely at different depths. You take another SIMS picture, and then you don't have to correlate with fluorescence. You can immediately compare, let's say, the turnover of these cells with the ratio of N15 to N14 versus the location of the specific marking in the endoplasmic reticulum. This is very nice, very easy to do. These are some later experiments where you now use the fluorine. You now label antibodies with it, and the antibodies then find a specific population of organelles. In this case, this is peroxisomes, and you can see them very nicely within these cells. Up here, this was another labeling method. If anybody's interested, I can explain it. This is called click chemistry. It's a bit more complicated, and it's only efficient in cultured cells. So with antibodies, you can do everything you want. They're very easy to use. You can also use N14, N15, I'm sorry, N14 is a naturally occurring isotope. You can use N15, which is the rare isotope, and you can put it on your probe, and then you have the ratio between N14 and N15 will tell you where your object of interest is, and you can even have sort of a semi-quantitative measurement of how many copies of that element you have there. So this is a bit of an advantage. It's a more noisy probe, but which is more quantitative. Albeit noisy, it has a better quantitative presence. You can use boron. Boron, you can see it here. You can label specific elements in the cells with it, and you can very nicely compare it with other elements in the cell. the good point about using boron is that boron is a positively, it's a, you know, boron is a positive ion. So so how should I say, ionizes positively to boron plus. And then, you know, things like N15 or fluorine, these ionize negatively. You can look at them in the negative mode together with oxygen, carbon, nitrogen, etc. Boron, you can combine it with sodium, calcium, magnesium, you know, you name it, and you can start looking then at these cells and start looking at the metal, for example, ions in there, which is very, very interesting. In this case, you would use the oxygen source to ionize. In the previous case, you would use the cesium source because that generates the negative ions. So what else can you do? You can do two color. So you can do multiple colors. You can use fluorine for one sample, and you can use iodine for another sample. By sample meaning, you know, like a different organelle, for example. In this case, we had iodine on mitochondria pieces and fluorine on peroxisomes, and you see them labeling the different organelles in these cells. You know, this, which looks very, very interesting. The iodine is not published. You know, we just recently made this probe, and I hope it's going to be available at some point soon. So what else can one do? Well, we had a bit of an issue with all of these probes that I showed you so far, you know, that they are all things that we made. So a chemist has to sit down in the lab, make them over some months, you know, and it's a lot of work. So in the end, we decided, why don't we just search the literature for and the markets for things that people can just buy, put on their sample, and immediately image it, you know. And fortunately, this is indeed possible. And what you can see here are different antibodies. They are labeled with different types of gold in this case. You have 6 nanometer gold, 15 nanometer gold, you have 1 nanometer gold, you have nickel NTA, which is a different type of labeling that binds gold directly, and you have even nanobodies directly labeled with gold, nanobodies being very small miniature antibodies. Not all of these things work as well as any others. I would say the 6 nanometer gold is probably the best. The 15 is so-so, and the 1 is not too bad. I mean, it kind of works. And the 1 nanometer gold also has fluorescence on it when you buy it from various companies. So this is probably quite advantageous because you can do fluorescence and SIMS at the same time. But the 6 nanometer gold is very nice, and you have here different organelles that you're supposed to label with gold, and you see gold labels for every one of them, and they are actually quite reasonable. I mean, you know, it's typically it's the shapes and the types of spots that you would expect, and they are actually quite satisfying. So finally, there is a tool that allows you to label in NanoSIMS exactly like you would label in fluorescence microscopy with commercial gold, you know, which you can just purchase and just can use. It's something that for us was quite satisfactory to finally get something that is easy to use and just from a simple commercial supplier. So, you know, I already spoke for almost half an hour. So I want to now dedicate the last few minutes to what else you can do. I mean, I hope I could convince you that you can feed your cells and look at things that happen over a long time, that techniques from the domain of electron microscopy or light microscopy couldn't really show you. But what else can you do? Is there something else that you could not do with other tools? And I think there are a few more points that are super interesting. And one is, you could just image other elements. You could image the burning of living cells. You could image their oxidation. You know, we all burn because we are all made of carbon and nitrogen-based molecules, and they get oxidized. You know, and we as humans, you know, we constantly get, our skin constantly gets rechanged. You know, why? Because it gets oxidized. And among the many oxidation reactions, you have reactions that just put oxygen within proteins. And then why don't you just image that? And this is something, it's not easy, it's not trivial, we haven't published it, it's not yet really working all as well as we would like it for publication, but it can be done. I mean, you can have your cells or your animals breathing an atmosphere of oxygen 18, and then you can go and look at it. You can go and see where that oxygen went. And there is no other technology that can show you this. So yeah, I mean, you can kind of see on the surface of this neuron, you see various patterns of where this oxidation apparently happens. And we still have to optimize this. We still have to do some biology with it, but it's essentially on the list. The other thing, and this is the last big point that I want to make, is you could image drugs. So one point that is important is, how do you image small molecules? Let's say you want to image a molecule that is very, very small in size. Let's say paracetamol, aspirin, ibuprofen, you know, something like that. You want to image them. How can you image them? This is an example. This is a drug that is very much used in a lot of diseases. I will show you. It's called ANLE 138B. You see it has one ring, a second ring, a third ring. So you know, a double ring here and two simple ones. It's tiny, 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 tiny. How can you put a fluorophore on it? Any fluorophore that you put on it will be actually bigger than the molecule itself. It will never work again. So what does this molecule do? You know, it was discovered by several groups in Germany that they collaborated, and they first described it as a molecule that delays damage in Parkinson's disease and in the Creutzfeldt-Jakob's disease. Then they discovered, hey, it also helps with Alzheimer's disease. So if you inject this into mice, mouse models of Alzheimer's disease live longer and happier. They also discovered it helps with another type of model of Alzheimer's disease. As you may be aware, Alzheimer's disease has different types of pathologies. Well, this drug helps with all of the ones that people looked at so far. It also helps with other diseases like multiple systems atrophy, which are diseases where things in our bodies sort of agglomerate, aggregate, and this drug stops them presumably from aggregating and from going bad, from becoming damaging to our cells. Unbelievably, it also may have implications for cancer. It actually seems to affect melanoma cells and to have some change in the fate of melanoma cells. So this is a very, very interesting drug. Now, we would love to know how it actually works. Of course, we have hypotheses, and of course, there are experiments in vitro made with ANLE, but how does it actually work? Nobody knows where it goes to in the brain. I mean, it probably has to be in the brain, but I mean, you can do a biochemical experiment and you know there is some level of it in the brain, but where in the brain, nobody knows. You can't image it. As I was mentioning, you can't put a fluorophore on it since it then is modified and it doesn't work anymore. So no small drug will work as a small drug if you put a fluorophore on it. That's I know of one example, so I'm lying. I know of one example that is a poison, a toxin. It's called phalloidin. That one binds actin and poisons you if you eat bad mushrooms, because it comes from a mushroom called Amanita phalloides. That one amazingly does work if you put fluorophores on it, but there are very, very few such examples. So perhaps you did notice that this one, that ANLE, has a bromine atom. So now you could image it in NanoSIMS, couldn't you? You could take your specimen, you could place it on your on your NanoSIMS, and then just look where ANLE is located, and that's exactly what you can do. I mean, these are here Cn minus ions. I'm sorry. This is then sulfur, and this is your bromine. So you can see that it accumulates in regions where your protein density is higher. You know, this is a collaboration with Christian Griesinger, a professor also in Göttingen at the Max Planck Institute. I'm not really allowed to tell you a lot more about this because we are ongoing in this work, you know, and it's not published, and it's you know, there are a lot of people interested in it. So I can't really tell you a lot more details of what we are seeing. But long story short, this is a method, or the method, to see where these small things are going. And there is a huge amount of drugs out there. Let's say not all of them have a bromine atom, of course, but then just go with you can go with N15. You can generate your drug with N15 and image N15. You can generate it with carbon 13. You could put several carbon 13s here, then you could image them. So this is, I would say, the last point that I wanted to make for today. The NanoSIMS is a key to seeing small drugs in situ, in the real animals, in the real. Also, you can take human samples like blood samples or whatnot, and you could see those. And yeah, I don't see another technology they could actually do this. So that's then the conclusion. I would like to thank you very much for your attention. I would like to thank my group for their work, especially Katarina, who is managing our NanoSIMS instrument, Paola, Fangsha, Elisa Bonin, also here, whom I forgot to write, and some of our biologists that work with NanoSIMS, like Sebastian and Sven. Also, great thanks to New, Sinem, and Ingrid, who are alumni of our NanoSIMS group, and New is the professor in Gothenburg, Sinem is a group leader in Heidelberg, and Ingrid is now has an important industrial position in Saarland. So yeah, that being said, thank you again.